considering RNA DNA backbone stereochemistry. So on the picture to the right here, we see a representation of RNA. So we can see the first nucleotide upwards and the other nucleotide downwards. And the arrows represent the RNA chain continuing indefinitely in both directions. So the phosphate group is not shown explicitly here, but implicitly. And but you know that nucleotide without the phosphate group is called a nucleoside, but it is shown here implicitly, so we consider this as two different nucleotides in an RNA chain. So what we're considering here is a thought experiment of the regiochemical selectivity of the chiral carbon atoms, which we display here in green. So you can see the first carbon atoms, there's a hydrogen atom attached and a nucleobase, and also a second chiral atom with also two different side chains that could be, we can imagine that the hydroxyl group being bonded on the other side and the hydrogen vice versa. So, and that holds for the third carbon atom where we can also imagine these side chains being bonded in different, in vice versa, and also for the fourth carbon atom. So, and continuing, and that's just considering this ribose moiety. But that holds for each nucleotide added in the RNA chain. So even in the second nucleotide, we can see that the first carbon atom has a nucleobase and a hydrogen that could also be switched in this direction. But there is very high fidelity in living systems that these are connected in this order in RNA and DNA. And but in DNA, in the second, there's no hydroxyl group, just two hydrogens in the DNA. That's the only difference, except for the difference of uracil being thymine in DNA. And so we can see that also there are 4 of these chiral carbon atoms in these two nucleotides. So you can count them just for 2. There are 8 of these chiral centers. You can imagine it when calculating combinatorial complexity, we just multiply each of these carbon atoms in green with 2, because there are 2 possible orientations at each of these carbon centers. So performing the calculations explicitly, considering 3 nucleotides, where this is a representation of 3 nucleotides, and so what we see here, because in these 3 nucleotides, it would be, if I point to the carbon atoms explicitly here that we're considering, we have just point them here explicitly. 1, 2, 3, 4, and second nucleotide on the ribose, 1, 2, 3, 4 chiral atoms, and on the third nucleotide 1, 2, 3, 4 chiral atoms. So then we have in this expression we have 2 for each of the 2 orientation of each carbon atom, and we have 3 nucleotides times 4 of these chiral atoms in each nucleotide. So then we will have a number 2 raised to the 12th, and this equals 4096, which is approximately the same as 10 raised to the 3.61. So there are, if we would have all of these in random orientations, just considering the 2 possible side groups of each chiral atom, not considering if there would be like other chemical groups competing for the same positions or any of the other many different kind of chemical compounds one could imagine being bound to each chiral atom, but just considering the 2 that are natively in RNA and in those positions. So then it's just there's a chance of 1 over 4096. So that's a very small number, basically 0, but it's so we understand that it's very unlikely to, if it would just be by chance, that this would be orient in that combination for just 3 nucleotides. And you can imagine the average size of nucleotides in RNA is about 500 to maybe 2000 nucleotides. So this is very slow. I mean, very little number, 3 nucleotides, but still we get a very unlikely chance of this ever happening by chance alone. So if we consider 4 nucleotides, we have the same calculations, but in this expression it will be 2 for each 2 combinations of each chiral carbon times 4 nucleotides times 4 of the chiral carbons of each ribose molecule of the nucleotide. So this will be the same as 2 raised to the 16, which is equal to 65,536, which is approximately 10 to the 4.81. So here you see that it's even more unlikely that this would ever happen by chance, and that so if we then consider 600 nucleotides, which is actually quite a bit, I think, lower average of RNA size when considering messenger RNA at least, and so 600 nucleotides. So we take 2 times 600 nucleotides times 4 chiral carbons centers of the ribose molecule, which is then 2 raised to the 2400, which closes into infinity, one could imagine. But the number is explicitly 10 raised to the 7, 222.47. And so this is just basically impossible that this would happen by chance. So there has to be some way how it could be arranged so specifically, RNA, and that raises questions where this specified complexity can arise from, and because this would never happen by chance, just considering these thought experiments of just these 4 chiral carbons in these RNA molecules, not considering all of many other complications due to competing chemical prebiotic chemical compounds that could be relevant in a prebiotic scenario. Considering the same calculation for DNA, which is deoxyribonucleic acid, which means it doesn't have these oxy groups on the second carbon position of each nucleotide. And that means there are two hydrogens bonded to the second carbon, so that make this one not chiral anymore. The second carbon is not chiral anymore. And that's why it's called 2-deoxyribonucleic acid DNA. So the overall structure one can see to the left here with a representation borrowed from PDB 101, molecule of the month DNA. And in this representation, they just have at the bottom one of the strands in red and the other one in green. And the top representation is atom coloring, similar to the coloring in the right representation of DNA. I think the white here occludes the black carbons, I think. Because there are a lot of hydrogens on the DNA molecule, and for human DNA, this one strand is 3.2 billion letters about, and each strand is 3.2 billion letters. And we can then consider a calculation of the entire human DNA molecule, because then we would have two possible orientation of side groups attached to each chiral atom, which is 3 rather than 4 as which RNA, but here it's 3 for DNA because of the rationale we used here to the right. And considering this for one strand of the DNA, which is 3.2 billion nucleotides, which is about 10 raised to 2.89 billion. And this is a very large number. We could approximate it with infinity for this matter of discussion at least. And because, calculating that this would ever happen by mere chance is basically 1 over infinity. It is closest in, it is 0, very close to 0, so that would never happen by chance alone. So there needs to be some kind of explanation to why this kind of preciseness and high fidelity in living systems can occur with DNA and RNA. Please write in the comments if anyone have good explanation and rationale for this. I haven't been able so far to find any good prebiotic explanation to how this kind of fidelity cannot occur in living systems